today you're going to be doing the DNA Electrophoresis lab. The first thing we'll do is look at some of your equipment setup. This is your flash gel, where your gel bed will go. These are your power supplies. We're going to go ahead and adjust those to as close to 275 volts as we can. You'll change that by rotating this knob. That's a little too low, so we'll turn that up a bit, and that's about as close to 275 as we can get. This one you adjust with these up and down buttons here, so we can get that right on 275. Once you get your gel loaded, I'll show you how to hook that to your power supply. Alright, so I'm wearing gloves because you're working with DNA. Your DNA samples are going to be in these microcentrifuge tubes, and notice that a lot of the DNA is up on the edges, so you may need to centrifuge your DNA down, and this spins it around so that all of it will get at the very bottom. So you may need to do that for a minute or so, or just a few seconds will be fine. Okay, so when you get this out, your DNA should be toward the bottom. Okay, so again, you're going to wear gloves, and I'd like for you to remember which lid goes with which tube. Okay, so you have a lot of holes on your setup here. You may want to spread them out so you don't get them mixed up. Okay, so you know which lid goes with which tube. The next thing you're going to use is a micro pipette. You'll need to select it to 6 microliters. Okay, so the way you do that is you unlock your adjustment here, and you can turn that. That's 5.5, and that is 6 microliters. You'll lock that into place, and this is where you hold the micropipette with your finger, and this is your plunger. When you're getting your DNA out, you're going to push one time, and you'll use a new tip every time you get DNA out. So you get a new tip on, push down one time, and you'll get your DNA out. You'll see a little bit of it in here, and then you'll push it twice to let the DNA out. I actually have forgotten to get our gel bed out, so let me do that real quick. This is how your gel will look when you open it. Be careful with these, very costly. You can just go ahead and lay that here and remember what DNA you put where. Okay, so you're going to hold this as such. I like to think of it as kind of like a pool stick. You're going to keep it steady with one finger, and you'll plunge once, twice, 2 plunges to get all that DNA out. And then you'll come over here and you'll use this button right here to eject the tip off. Okay, so you'll get a new tip, and then you're coming in your second DNA sample. Plunge it down once, bring it up, and then you're going to come in your second lane. 1, 2, and you'll eject that tip. Then you'll load up your third sample, plunge it once, down in here, out, and plunge it down twice. 1, 2, eject your tip, and get you a new tip, and you'll get your next sample, and bring it over here, loading this up. In this particular experiment, you will use 6 DNA samples. Okay, I'm actually chose some different ones other than what you guys are doing because I didn't want you to see the exact results. But y'all's will have standard, mother, child, father 1, father 2, and we have room for you to do 1 practice run. Okay, that's what these colors represent. Okay, so this is your standard. It will have several DNA lines that show up. Okay, and then you'll have mother in your next well, and so on. Child, this is father 1, father 2. In your orange, you can see we have a lot of it. That is your practice. Okay, so those will go, those 6 will go in these 6 lanes. 1, 2, 3, 4, 5, 6. Okay guys, you're actually going to share this particular gel bed with a second group. So another one of the groups in your class will use these 6 lanes. Standard, mother, child, father 1, father 2, and practice. So you may want to do your practice first. Okay, so your group will be using this as well as another group. Once you get your DNA loaded, you're going to put that into your flash gel chamber. As such, there's really only one way it'll fit in there. Okay, and then you're going to plug this up to your power supply. Either one of these work. You just simply hook the black to the black and red to the red. So we'll use this one, and we'll put black to black and red to red. Okay, and this will begin emitting electricity to the DNA. DNA is a polar molecule, so it has a positive end and a negative end. In this particular experiment, in any DNA electrophoresis, the negative end is here. Okay, and so what's going to happen is the DNA will move very quickly to the positive side. And so as we look down right here, you can probably see bubbles. Actually, it's bubbling up right there. So you can see bubbles on this electrode. So that means there's electricity running through the DNA, and depending on the size of the fragments of your DNA, some move faster, some move slower. So the smaller fragments move faster. So you will begin to see your DNA fragments move along these lanes. It shows up really well with your lights turned down. So we're going to turn our lights down now, and then you'll be able to see your DNA moving just a little bit. You'll turn your UV light on here, and you'll see the DNA, let me find a pointer, start to run. Okay, so it's moved from the well where we loaded it, and these 4 are moving down the lane. Now, the UV light actually degrades the DNA, so you're going to let the sample run for about 8 minutes, as long as it takes. And so you'll turn it on in the beginning to make sure that it's working, and then turn it off. Okay? And then you'll check it again, let it run with the light off for about 4 minutes. Okay? And then turn it on again at 4 minutes. And during that time, take a ruler and put your ruler on your results chamber here. This is 0, of course, and you can put a little piece of tape so your ruler will stay in position. And then you can actually measure how far each of the DNA fragments are moving. And you can see this one has 1 DNA band here, and it looks like this one and this one may have a couple of more that are beginning to show up. Okay, so we're going to turn that off and let it run a little while longer, and we'll look over here at your results while that's running. So for example, you're trying to figure out which father is the father of the child. So for instance, in this example, you can see that this is the mother of a child, and this is a potential father, megabucks. So the child has this DNA band. Notice it didn't come from mother, but if you follow it down from where it was loaded, it matches up with the same line of that father. This particular band on the child did come from mother. This one from mother. This band mother doesn't have, but if you look over here, matches up perfectly with that potential father. This one came from mother, same way with here. This one came from that father, that came from mother, that came from mother. Mother doesn't have this one, but father does, and that one's the mother. So in your sample, you'll be looking at the child, and you'll be matching up which bands belong to mom, and trying to see any band that the child has, which father that it came from. Okay, so as you can see, after a few more minutes, your results have migrated further. What I recommend you do, because the results don't last very long, I recommend that you take a picture of your results when you get them like you like them. and you'll have them here. These are just some you know, sample results that are going to be a little different from yours, and you'll draw how far they migrated down. So as you can see, we're at about 1.5, 1.6 centimeters. So on your results paper, it tells you to draw the results, and so you'll begin, you know, you can mark that in centimeters and millimeters down here, and then kind of draw your bands for each of the people that you had a sample for. And when you're finished, of course, you can throw away your tips, and you'll put your ruler back in the bag for rulers, and then, you know, once you throw away your tips, you will put your empty tray back into the used Petri dish bag. Okay, thank you.